My name's Troy Brier. I'm a member of the Luthi-Schulten group here at the University of Illinois at Urbana-Champaign. And today I'm going to discuss um, the genome architecture in Syn1 and the minimal cell Syn3A. And so to get started, we've talked about these major cellular processes, or people have talked about these, my colleagues earlier, the idea of the genetic information processing and the metabolism. And so this is how the cell is able to go from the genotypic understanding to the phenotypic understanding. And we have these cellular processes, and we like to describe them based on their states. And the way we describe their states is based on the counts or the numbers of species within those subsystems or those systems. And so genetic information processing can be broken into three component levels: the genome, composed of the DNA, the transcriptome, composed of the RNA, and the proteome composed of the proteins. And then your metabolism system can be described by its components, a part of the metabolome. And so what I have at the bottom here are some numbers from Syn3A that give you a rough idea each of these components, how much each of these components are with respect to the mass of Syn3A, and then also their abundances. Again, we describe these components or the states of these components by the numbers of the species involved. And so what you're all probably familiar with is that all of these different component levels are connected through different processes. So I have some of the major ones depicted here: transcription, translation, and enzymatic reactions. But what's also important to note, and I don't have this shown, is that these levels going from genome to metabolome can impact each other in kind of this direction of genotype to phenotype, but there's also feedback control at the metabolome level, let's say in the precursors, like let's say the nucleotides being used to construct the genome or the genes. And so what I and my group has been kind of focused on with some of our simulations is trying to understand how these different levels impact each other. And one specifically that we're trying to look at is how the genome impacts the rest of the component levels of the cell. And so there's been some work that's already talked about how these factors at the genome level influence the other component levels. And so my colleagues earlier, they talked about these first two or touched on them, or this first one, I should say, whereas the actual physical orientation of the 3D chromosome seems to impact the messenger RNA half-lives, which would obviously impact the transcriptome and so on downstream. The other one that our group has also looked into is the gene loci along the chromosome. And so throughout the cell cycle, your gene copy number will vary. And so what that results in is, if you look at this graph on the right, is that as your gene copy number varies, you also have your mRNA copy being able to vary and enter in from a high or a low state to a high state. And so that's based on the fact that if you have two genes present, they both can be transcribing messenger RNA versus a single gene. And so what those states or entering that state is based on your time of replication for that gene. And that is directly related to where the gene is on the circular chromosome. And so the first two I just touched on, they focus more on spatial aspects of the genome. These other two that I would like to touch on and expand upon deal a little bit more with the sequence aspect of the genome. And so one way we know that the genome definitely impacts the other levels is all of the functions of the cell are encoded within the genes. But this last bullet point that I would like to touch on is the idea of the genome architecture, the local arrangement of sequence motifs. How does that impact these other levels? And so what I'm showing here is the ATP synthase structure. And what I would like to point out is that the structure has multiple subunits involved in it, and all of them in a different stoichiometry. But what is very interesting is that if I show you the genes where they're located on the circular chromosome, so they're all right next to each other. So that would kind of get rid of these first two spatial components because those impacts should be roughly about the same due to the fact you expect these genes to copy about the same time, and locally they should be roughly in the same region. But what's interesting is the idea is how can this genome architecture be able to generate these different stoichiometries or ratios of the protein species needed to make this complex. And so one way to describe the genome architecture is to look at the genome sequence motifs or features that are on the genome. And so I've touched on this idea already, is the coding region. So that codes for the mRNA, tRNA, rRNA, or small RNA. But there are some other additional motifs that are important and seem to have impacts at the other cellular levels. So one of them being the Syn1.0, where I have the template sequence drawn out there. And that's based on an interaction between the 16S 3' end of the 16S rRNA interacting with the messenger RNAs, and that's been shown to enhance translation initiation. There's also the promoter sequence, which I'm sure many in the audience are familiar with, and we have a template sequence for that. And so the important thing to note with the promoter is that this is, you can think of this as the start of transcription. There's also the transcription start site, which is where transcription, or where the DNA actually begins to be transcribed into messengers. There's also the intrinsic termination loop, which is the important sequence that, or important motif that denotes the end of transcription. And so in Syn3A and other gram-positive, this motif becomes more relevant due to the fact that most of transcription termination is believed to be row independent. So it does not use a protein to terminate transcription. There are also some other sequences that were touched upon. I'm not going to go into these too much during this talk, but an example of them would be riboswitches, and also the DNA signature, which Zane touched on, is how replication is regulated or controlled in Syn3A and other related organisms. And so now that we have identified these sequence motifs, the big question to ask is, how are these motifs arranged on the circular genome? And so to determine that, what you can do is identify each of these sequence motifs along the genome, and then using their coordinates, you'll have an understanding of where they fall along the circular genome. And so the first thing that we can start is looking at the open reading frames, which are defined in the GenBank file for both Syn1 and Syn3A. This is the easiest step. The following is then to use related organisms, or information from related organisms where similar studies have been done, to identify these other motifs. So the Syn1.0 was identified based on the spacing region between the gene open reading frame and the Syn1.0, and that was identified using a sequence alignment-based approach to determine the best potential motif. And then continuing from there, what you can then do is, now that you've identified a potential Syn1.0, you can then identify the other features. The transcription start site is not necessarily defined computationally, but what we can do is use, again, related or information from related organisms to define a search region for the promoter. And so the promoter was found in a very similar way to the Syn1.0. Then the very last thing is to find that last motif, which is the intrinsic termination loop, which was done using a decision rule that was calculated based on the RNA secondary structure and the T-stretch region downstream of a gene. And so this was done, this search region is a little bit more dynamic in that it's done from the end of a gene until 500 nucleotides, or the beginning of another gene, to not allow for significant overlap. And so I touched on the idea that the promoter and the intrinsic termination loop define the boundaries of transcription, the start and the end of it, which is how all information from the genome is converted to the next level, the transcriptome. And so what we can then do is use this genome architecture as it's defined by these sequence motifs to define transcription units. And so this is done, and this figure is adapted from some work from Sebastian Rodriguez's lab, which he'll be talking, I believe, on Monday. Or next Monday, I should say. And so what was done here is you can then add in the term prediction, add in the promoter predictions, and define these transcription units. And so an example would be this region here, where you have transcription unit 1, which encompasses gene B and C, and then you also have transcription unit 2, which encompasses just gene C. And so this idea allows you to kind of understand how potentially co-expression events can be described in an organism. And so doing this in Syn1 and Syn3A was we found this to be a useful kind of endeavor due to the fact that we know there's not a lot of regulation in this cell, and so potentially these impacts from the genome architecture can be better understood in these organisms compared to others. Um, with those computational predictions, we then needed to find a way to verify them. So some experimental predictions were done. And one way, or they were done using transcriptomics. So it was done using Oxford nanopore long read, where based on this figure on the right, kind of goes through the idea of the experiment. You have some RNA prep stage. It's important to note that the rRNA was removed in these studies to make it easier to better understand the mRNA and tRNA due to the fact that they're at significantly lower levels comparatively to the rRNA. And then that RNA prep is fed through a membrane nanopore to be sequenced and determine where it falls along the genome. And so those sequence reads are processed and aligned to the coding regions, which is the CDS and the tRNA genes. And then these alignments are processed into transcription units. And so now we have a roughly one-to-one comparison between computational approach and experimental approach to identify these transcription units. And so I'll quickly go over some of the results of these studies. So one thing we're very happy to find is that looking at the comparison of the computation to experiments, so this light blue to the red, there's pretty good agreement between the number of genes within a transcription unit. The averages, as you can see, are 2.5 for the computational and 2.9 for the experimental. It's important to note that the computational, as you can see, there appears to be more transcription units identified, and that can be seen on the table to the right. And that has to be that's due to the computational method allowing for a less static picture of the genome, where every single promoter and transcription or intrinsic termination site is allowed and assumed to be able to be a transcription unit, whereas the experimental data is based purely on what is observed in the experiments. Another kind of nice thing to note is if you look at this column to the right, a similar type study was done in Mesoplasma forum, where they were able to identify the transcription units using transcriptomic analysis, but an analysis that differed than the one that I previously discussed. And what's interesting is that although a different experimental method was used, the computational predictions still fall in very good agreement with the experimental results. As you can see, based on the length of the transcription units, the number of genes, and then even in the numbers. And then interestingly enough, the longest transcription unit found in all of these actually belongs to a set of genes, a set of ribosomal proteins that are organized within the genome in a quite long transcription unit. And so with that, I would like to conclude. And so some conclusions I would say we've been able to pull from this work is that the gene sequence motifs are organized along the genome and are identifiable using sequence-based searching methods. Using the boundaries of transcription, promoters, and termination loops, transcription units were identified in agreement with Oxford nanopore sequencing experimental results. And then some ongoing work we would like to do with this is John and Ben earlier touched on this HUP-A, or the histone-like protein. So what we've been able to identify is that in the minimization from Syn1 to Syn3A, the genome architecture of that gene has been altered. And so we would like to see how those predictions impact or how the minimization impacted the genome architecture and thus the expression of that protein. The last couple points are to collect some experimental data on Syn3A for comparison, because we do not currently have experimental data from Syn3A. And then the last two bullet points can kind of be conjoined together, but just the idea of trying to generate an expression model using these transcription units to better understand co-expression, but understand how this genome architecture plays a role in the expression and thus the other levels of the cells. So by levels, again, the transcriptome, proteome, and metabolome. And with that, I would like to acknowledge my advisor, the rest of my group members who you heard speak earlier, the JCVI synthetic biology group, specifically John Glass, Kim Wise, Clyde, and Ham, for all of the help that they've given. Additionally, also Andras and Daniela, And then I'd also like to thank Dr. Pratap Benpali, who did the experimental transcriptomic analysis. And with that, I'll take any questions if we have time for that.
Roy, what wet science would you wish for that you don't see on the horizon, at least my group doing immediately? What would make this significantly more relevant as an explanation of how cells do this sort of thing?
So a nice, or one thing that I would like to see or be able to look at is, so there are some additional transcriptomic analysis methods using techniques like MERFISH out of the MOFIT lab. And so to get a better quantitative description of how how the transcription unit architecture impacts expression. And so a way to do that would be potentially you could know what your promoter sequence, know what your Shine-Delgarno, basically know what the motifs are going in, put that into, let's say, the landing pad of Syn3B, and then use this MERFISH technique, which is a super resolution imaging technique, to be able to quantitatively get at the numbers of the transcriptome and proteome based on these genome architectures. And then kind of we would be able to use simulation approaches to better describe or understand what those impacts are directly are. I'd also like to, I guess, note the idea that we're not necessarily saying that transcription units are the reason why all of these things are happening. It's just we think these are one of the factors that are playing a role into how the cell's genotype is developing or evolving into a phenotype.
So maybe you and I should approach the MOFIT lab and see if we can, get them to join our
Yeah, that would
program.
be nice.
Anyone else?
Hi, I'm Veronica. I
Hello.
was wondering if you see these high expressing transcriptional units close to the ribosomal RNA, because you said that they were pretty highly expressing. And in work from E. coli, it's been shown that high transcriptional propensity is and somewhat dependent to the proximity to rRNA operands.
So I haven't looked directly into that. Due to the fact that the RNA is not in the experimental data, I don't look at it as much in terms of worrying about its transcription unit architecture. But that idea of looking at genes near the RNA is something that I will definitely look into. One of the things that we're kind of just interested in general is how much of, as I kind of said, but how much of the genome that's in, or how much information that's encoded in the genome can we pull out and be able to understand what it's going to evolve into downstream. So I was unaware actually of that relationship of proximity to the rRNA gene. So thank you for that, and I will definitely look into that.